All right, let's just dive right into this explainer. We're about to embark on an absolutely incredible journey through the actual mechanics of life. I'm talking about scaling all the way up from the microscopic folding of a single tiny molecule right up to the massive global dynamics of entire ecosystems. So if you've ever found yourself wondering how those invisible chemical reactions happening inside your cells right this second actually relate to the survival of our entire biosphere, well, you're exactly in the right place. Let's get into it.
To keep us on track, we're going to use this agenda as our mental map. We'll start with the molecules of life, move into human genetics, then anatomy and reproduction, tackle animal behavior and homeostasis, and finally wrap up with population ecology and conservation.
Which brings us to Section 1, the molecules of life. Okay, so it all comes down to this beautifully simple sequence, the central dogma of molecular biology. Basically, everything starts with DNA, right? It acts as the ultimate vault for your genetic info. But the thing is, DNA stays locked up tight in the nucleus. It doesn't leave. So an enzyme called RNA polymerase steps in. During transcription, it copies that DNA into messenger RNA or mRNA. That mRNA is essentially a delivery system. It carries the code out to the ribosomes where translation kicks in, synthesizing the actual proteins that build and run your entire body.
Now, what's truly wild is how these proteins, specifically enzymes, act as these hyper-precise biological regulators. They don't just blindly speed up reactions, they're incredibly controlled. Take allosteric enzymes, for example. They literally have additional binding sites. When an effector molecule parks itself there, it can basically flip the enzyme's catalytic activity on or off. We also see regulation through covalent modifications like phosphorylation. Honestly, it's like an intricate microscopic switchboard, just ensuring reactions only happen exactly when they need to.
And when substrates actually do bind to these enzymes, we rely on two pretty famous kinetic models to explain what's happening. You've probably heard of the older lock and key model, which assumes the enzyme's active site is super rigid. It has a specific geometric shape, like a key fitting perfectly into a lock. But modern biochemistry leans heavily toward the induced fit model. It's so much cooler and far more dynamic. In this model, the active site actually changes its shape to give the substrate a perfect custom fit upon binding. This insane level of specificity is exactly why lactase only breaks down lactose and why DNA polymerase is so perfectly suited to copy DNA.
All right, zooming out a bit, let's hit Section 2, Rules of Human Genetics. So you might assume that one gene just flat out dominates another, right? We're all taught those foundational Mendelian laws, segregation, independent assortment, which make it seem like there are always clear dominant and recessive traits. But as we know, biology absolutely loves nuance. So what actually happens when things aren't so black and white?
Well, the crucial takeaway here is that genetic traits can actually share the spotlight. Take codominance. This is where both alleles are expressed equally in the physical phenotype. Picture a flower with distinct, bold red and white patches. Neither one is hiding. On the flip side, we have incomplete dominance. This is where the traits literally blend together, creating an entirely new intermediate expression in heterozygotes. So instead of patches, that red and white flower produces a totally pink offspring. Pretty neat, right?
And really grasping these rules is absolutely critical when we start looking at human health. By tracing vertical parent-to-offspring lines on a pedigree chart, we can map exactly how conditions get passed down the family tree. For instance, hemophilia, which is a disorder where the blood fails to clot normally, that's passed down via sex-linked inheritance on the sex chromosomes. Conversely, autosomal recessive disorders are a bit different. They require you to inherit two defective genes, one from mom, one from dad. Trying to map these super complex patterns across thousands of data points is literally why the modern field of bioinformatics, using powerful software to crunch biological data, is so incredibly essential today. Okay, now that we've mapped out the genetic blueprint, let's step up to Section 3, Human Anatomy and Reproduction. Think about this for a second. Every single movement we make, from blinking to running a marathon, is just a crazy cascade of microscopic chemical reactions. It starts with an electrical action potential that triggers the sarcoplasmic reticulum to flood the area with calcium. That calcium is the key that allows the two main muscle filaments, actin and myosin, to finally interact and form what's called a cross bridge. And of course, to keep this whole cycle going, ATP has to be constantly synthesized in the mitochondria to recharge the entire system. Now, depending on exactly how those actin and myosin filaments pull against our skeletal system, which, remember, is made up of long, short, flat, and irregular bones all linked by joints, our muscles actually perform three very distinct types of contractions. If the muscle shortens, that's a concentric contraction. If it's lengthening under tension, that's an eccentric contraction. And there's a third type,
An isometric contraction. That's when you're generating force, but the muscle length actually doesn't change at all, like pushing against a brick wall. But muscles aren't the only things driven by chemical cascades. Human reproduction relies on a meticulously timed, delicate chemical dance, orchestrated mainly by 4 key hormones. Throughout the menstrual cycle, follicle-stimulating hormone, or FSH, steps up first to mature the follicle. Meanwhile, estrogen gets to work building up the endometrial lining. Then, a sudden spike in luteinizing hormone, or LH, is what actually triggers ovulation. Finally, progesterone tags in to maintain that endometrium during the luteal phase, prepping the body for a potential pregnancy. If you disrupt this balance even a tiny bit, it can profoundly impact fertility. It's just a brilliantly complex physiological loop.
Let's step completely outside the internal human body now for Section 4, Animal Behavior and Homeostasis.
If you've ever watched bugs react to a sudden environmental change, you know they definitely do not all act the same. Like a woodlouse will just wander completely aimlessly when it gets too hot. But a cockroach? If you flip the kitchen light on in the middle of the night, it makes an absolute beeline away from that light source. So what's the deal with that?
Well, the difference really boils down to intent. Both of these are innate behaviors, meaning they're raw instincts hardwired into their genes, not something they learned. But kinesis is just a totally random movement in response to a stimulus. That's our wandering woodlice. Taxis, on the other hand, is a highly specific directional orientation. So that cockroach fleeing the light, it's displaying negative phototaxis. It's literally executing a directional escape plan written into its DNA.
Now, when we look at acquired or learned behaviors, we've got to talk about classical versus operant conditioning. Classical conditioning is involuntary. It's about associating a completely neutral stimulus with an unconditioned one. The classic example is Pavlov's dog automatically salivating at the sound of a bell. Operant conditioning, though, relies on voluntary action and consequence. That's when you give a dog a treat as positive reinforcement because it successfully sat on command.
But hey, knowing how to behave isn't enough to stay alive. Organisms also have to maintain intense internal balance, or homeostasis. Whether the threat is dropping temperatures or a massive spike in blood sugar, homeothermic animals use specialized receptors, control centers, and effectors to manage it. If you're freezing, vasoconstriction physically narrows your blood vessels to trap heat in your core. Sweating it out, vasodilation opens them up to cool you down. We see that exact same balancing act with blood sugar. Insulin steps in to store excess glucose as glycogen, while glucagon does the exact opposite. It breaks that glycogen down to elevate glucose levels when you need a quick burst of energy. For our final massive zoom out, let's apply all these rules to the grandest scale. Section 5, Population, Ecology, and Conservation. Look, to really understand a population, you've got to figure out how to measure it first. And ecologists use very specialized methods to gather these demographic parameters, depending entirely on what they're studying. I mean, you obviously can't use the same tool for a pine tree that you'd use for a pack of wolves, right? So for sessile, or non-mobile organisms like plants, you'd use quadrat sampling. But for animals constantly on the move, you have to rely on the mark recapture method, carefully trapping, tagging, and releasing a sample before trying to find them again.
Once they crunch that data, ecologists can visualize the age distribution using population pyramids. It's a brilliant tool because you instantly see a community's trajectory. If you see an expansive pyramid with a really broad base, you're looking at a high birth rate, a booming population. A constrictive pyramid with a narrow base, that population is shrinking. Meanwhile, a stationary pyramid tells us things are relatively even and stable across the board.
And these pyramids are what allow us to build serious mathematical growth models. In a totally perfect world with unlimited resources, a population will just explode in exponential growth, shooting straight up in a dramatic J-shaped curve. But, reality always fights back. Thanks to density-dependent factors, things like disease or just competing for food, populations are forced into logistic growth. That's the S-shaped curve, where growth eventually slows down and levels off right when it hits the environment's maximum carrying capacity. And when populations hit those limits, especially us humans, our activities inevitably put massive stress on the environment. Rapid urbanization and industrialization causes severe depletion of non-renewable resources. Then there's the terrifying reality of bioaccumulation, where harmful toxins just keep building up in organisms as you move higher and higher up the food chain. This is exactly why modern conservation efforts aren't just nice to have, they are absolutely crucial. Establishing strict nature reserves in botanical gardens, like some of the really impressive modern mechanisms they're implementing in Ethiopia right now, is critical if we want to preserve biodiversity against this rapid depletion.
And that actually brings us to the end of this explainer. We've gone all the way from the microscopic folding of a tiny allosteric enzyme right up to the vast, complex ecological carrying capacity of planet Earth.
And it is all so deeply interconnected. Because honestly, the true beauty of biology isn't just memorizing facts off a whiteboard. It's realizing that the chemical reactions firing inside your cells at this exact moment are following the literal same laws of nature that govern the survival of our entire biosphere. So the next time you step outside, I really want you to ask yourself, how are you going to use these rules to understand the living, breathing world around you? Keep questioning, keep exploring, and I'll see you next time.